Today we'll learn about steps of thyroid hormone synthesis. So in this first step is iodide trapping. So this iodide, it is trapped inside the follicular cell cytoplasm from capillary blood, and the channel required here is sodium iodide symporter. There occurs co-transport of 2 molecules of sodium and 1 molecule of iodide inside the follicular cell. And this is a kind of a secondary active transport for which the energy required is provided by the sodium potassium ATPase pump. Now, the next step is when this iodide comes inside the cell, it must be oxidized before entering into the colloid. So this iodine, it is oxidized into iodine in the presence of H2O2 and thyroid peroxidase enzyme. And this oxidized iodine enters into the colloid with the help of chloride iodine anion exchanger, which is also called as a pendorine. So this oxidized iodine enters into colloid of follicle of thyroid. Now the next step is formation of thyroglobulin molecule. The thyroglobulin, it is a glycoprotein. It is synthesized inside the follicular cell endoplasmic reticulum, then packed into Golgi apparatus, then in the form of vesicles, it enters into the colloid by the process of exocytosis. So first step is iodide trapping. Second step is oxidation of iodide into iodine. Third step is formation of thyroglobulin. Now this thyroglobulin is present inside the colloid, and this thyroglobulin mainly consists of tyrosine amino acids on which this oxidized iodine is going to attach. This oxidized iodine, if it attaches, one iodine attaches with the tyrosine residue on the thyroglobulin molecule, there is formation of MIT, that is monoiodotyrosine. If the two molecules of the oxidized iodine attaches on the tyrosine residue of thyroglobulin molecule, then there is a formation of DIT. This process of attachment of oxidized iodine with the tyrosine residues of thyroglobulin is called as organification of iodine. After this organification of iodine, there occurs coupling reaction, where this MIT, if it couples with DIT, there is a formation of T3. And if DIT couples with DIT, then there is a formation of T4. So, because of this coupling reaction, there is a formation of T3 and T4 thyroid hormones. And sometimes when the coupling do not occur, the MIT and DIT, they remain as it is on the thyroglobulin molecule. So, thyroglobulin molecule, it is stored inside the colloid of the thyroid follicles, and it may contain MIT, DIT, T3, T4, and sometimes RT3 is also formed. So, it is stored, this thyroglobulin along with the thyroid hormones, it is stored inside the colloid of the thyroid follicle. And whenever required, whenever there are signals are there, then this thyroglobulin molecule along with the thyroid hormones, it is taken inside the follicular cell by process of endocytosis. And inside the follicular cell cytoplasm, the lysozymes acts on this thyroglobulin molecule along with thyroid hormones, and because of this lysosomal enzymes, there occurs proteolysis, and this thyroglobulin along with thyroid hormones, it is converted into T3 and T4, which directly enters into the capillary blood. And MIT and DIT, they do not enter into the capillary blood, they are deiodinated by the deiodinase enzyme, and they are converted into iodide, which is recycled. So in this way, the thyroid hormones enters into the circulation. So these are the steps of thyroid hormone formation. First step is iodide trapping. Second step is oxidation of iodide to iodine. Third step is formation of the thyroglobulin molecule. Then fourth step is organification of iodine. Then there is a next step is coupling reaction, and the last step is proteolysis of this thyroglobulin molecule and release of T3 and T4, and MIT and DIT, they are deiodinated and converted back into iodide, which is recycled. So that's all about thyroid hormone formation. Thank you.